Assalamu alaikum. Welcome to this session. Today I am going to discuss about the processing of RNA, that is the post-transcriptional modification of RNA after their synthesis. DNA polymerase of replication has a unique property of proofreading, that is the daughter DNA that are synthesized from the base sequence of parental DNA. Any error that occurs in this DNA that are identified and removed by DNA polymerase. But the RNA polymerase has no such effective proofreading property. So the RNA molecules that are synthesized during transcription are modified post-transcriptionally. All three major types of RNA, that is the messenger RNA, transfer RNA and ribosomal RNA, undergo post-transcriptional modification, except the prokaryotic messenger RNA that is the same as that of its working copy. So it does not need any modification. But the eukaryotic messenger RNA needs extensive modification after their synthesis. This is the structure of messenger RNA. It has two ends. This is the 5' end, this is the 3' end. At the center is a coding region. It's a coding region or transcription unit. It consists of alternative arrangement of exons and introns. This is exon 1, exon 2, exon 3. A primary transcript of messenger RNA is the initial linear RNA copy that are synthesized from the RNA polymerase II, that is known as heterogeneous nuclear RNA, that undergoes extensive post-transcriptional modifications. These are the 5' capping, the addition of, the number 2 is the addition of poly A-tail. Then the removal of introns. The last one is the splicing. The first modification is the 5' capping. Here, a cap-like structure is added at the 5' end. It's a cap-like structure by attachment of 7-methylguanosine triphosphate that is attached at the 5' end by 5' to unusual 5' to 5' triphosphate linkage that occurs at the 5' end. And at the 3' end, there are several hundreds of adenine nucleotides are attached. These are known as polyadenylation. That is attached at the 3' end. Ultimately, these extra adenine bases are removed or excised, and these two modifications stabilize the messenger RNA, the newly synthesized messenger RNA, from its transportation from nucleus to the cytoplasm. So these two modifications protects the newly synthesized messenger RNA from the premature degradation of exonucleases by exonucleases. The third modification is the removal of introns. Exons carry genetic information, but introns do not, so they are removed. Sometimes some messenger RNA may not contain introns, but it may present as much as 50 introns that is present in the collagen, alpha chain of collagen. 50 introns may be present in the alpha chain of collagen that must be removed. Introns do not carry any genetic information, but they help in rearrangement of the genetic material, and it is helpful in evolutionary process. And it is not necessary that the exon 1 always reunites with exon 2, it may join with exon 3, and it is known as alternative splicing. And due to this alternative splicing, diverse proteins diverse protein molecules are synthesized from limited sets of genes. Removal of intron and joining of exons is known as splicing. That is, in between two exons, these introns are removed during the process of splicing. Like this. Suppose it is exon 1 and it's the exon 2. In between two exons, there is an intron that is to be removed. So it takes a look like this, and snipping occurs here, and also there. And ultimately it takes a look like a lariat or a lasso, just like this. That must be discarded. And the splicing is facilitated by small nuclear RNA. And small nuclear RNA is rich in uracil, and some protein molecules are also incorporated here to form small nuclear ribonucleoprotein, commonly known as SNRPs. And the whole complex is known as spliceosome, that is responsible for the process of splicing. Systemic lupus erythematosus is an autoimmune disorder. Here, antibody formation occurs against patient's own nucleoprotein, that is against SNRPs. The antibody formation occurs against SNRPs in SLE. Sometimes mutation occurs at the site of splicing, known as splice site mutation, that forms a wrong protein or aberrant protein that is found in some types of beta thalassemia. Over 15% of the total genetic disorder is due to this splice site mutation. The primary transcript of ribosomal RNA is synthesized from long precursor RNA molecule, that is known as pre-ribosomal RNA. It is the pre-ribosomal RNA, and it is synthesized as long precursor molecule that is cleaved by ribonucleases. And cleavage occurs here, and also there. And ultimately, it takes a look like this. 28 is, 5.8 is, and 18 is ribosomal RNA. Ultimately, it forms an intermediate sized desired RNA species. And it is further processed. That is the synthesis and processing occurs in the small nuclear RNA, snRNA, and it occurs within the nucleolus. And base and sugar modification also occurs here. And some protein particles, protein molecules are incorporated into the structure of ribosome prior to or during the post-transcriptional modification of ribosomal RNA. Ribosomal RNAs are transcribed by RNA polymerase 1, except the 5S ribosomal RNA, 5S eukaryotic ribosomal RNA, that is synthesized by RNA polymerase 3. Primary transcript of transfer RNA, that is the pre-transfer RNA, has extra long sequences, extra bases at its both ends, both in 3' end as well as in the 5' ends. It's the 3' end of pre-transfer RNA and is the 5' end. At the 5' end, near about 16 nucleotide bases are present and that are removed during post-transcriptional modification of transfer RNA. 16 bases may be present here. And initially, the 3' end contains uracil bases that are replaced by a CCA sequence to form the acceptor arm for attachment of amino acids, for synthesis of polypeptides or protein during translation. Some unusual bases are present at the D loop and T size C loop. It's the D loop and here is the T size C loop. Here the unusual bases are added and it is the normal characteristic of transfer RNA. The presence of this unusual bases is the dihydrouridine and pseudouridine that is present in the D loop as well as in the T size C loop, and it is the normal characteristic of transfer RNA. There are some introns, near about 14 introns are present in the anticodon loop. It is the anticodon loop that must be removed. So, after this modification, the pre-transfer RNA is transformed into mature transfer RNA. It is the pre-transfer RNA that is transformed into mature transfer RNA. Transformed into mature transfer RNA. Transfer RNA molecules are synthesized by RNA polymerase III as well as the 5S ribosomal RNA synthesized by RNA polymerase III. That's all about the processing of RNA after their synthesis. Thanks for watching. Assalamu alaikum.